All right, so next thing we're going to be talking about here is the histology of the small intestines. When we're really looking at what the small intestine's doing, it really is getting into kind of that food that we've started breaking down in the mouth, in the stomach. It's really now going to start trying to complete that, and this is chemically really trying to break down to the basic subunits of these nutrients and get them into their absorbable form. So proteins down to amino acids, sugars down to monosaccharides, fats down to their basic subunits, same thing with nucleic acids. Part of what we need to do in the small intestine is protect it from the contents of the stomach. The other thing is there are going to be some hormone secretions, and we'll see a little bit when we talk about the digestive accessory organs. We'll see a little bit about some of the things that they can secrete based on what these hormones do. If we're looking at this, we have a few different things going on here. Closer to the stomach, so the further end up here, if we want to call that the proximal end, it's going to have a lot more, what are these called, these plicae circulares. So these are circular folds that kind of go around that lumen. Their more colorful name is the valves of Endocrine, which sounds like it's some Lord of the Rings type of thing here. The valve, he went past the valves of Endocrine. So it's kind of a fun name on that one. We're going to see these are more common further up in the small intestine. As you get further down, these start to become less and less until they're not there anymore. What they are kind of doing is increasing that surface area of the small intestine. On top of these folds, a lot of times we will see these finger-like extensions called villi. These are lined with epithelium on the surface. It's going to be a simple columnar epithelium with microvilli on the surface there, and that is what is making up that mucosa. But these plicae circulares, or valves of Endocrine, are these folds that are both submucosa and mucosa going up and into there. Helps to slow the stuff down as well as increase surface area, like I was saying. We're going to see that there is three main regions to the small intestine. You have the duodenum, which is right after the stomach, that from that pyloric region down. We looked at it a little bit on one slide already. It's about the first foot. After that, we have the jejunum, which is the next 8 feet after the duodenum. And then the last and longest part is the ileum, that most distal region. And when that ends, that's where we're going to get into the actual large intestine and the parts of the colon. So what we have here is it's basically this 22-foot long or 21-foot long folded pipe with these folds on top of folds on top of folds. So overall, how much surface area are we really getting at? Well, this 22-foot long tube that's maybe an inch in diameter, it is giving us what would really be almost a tennis court of surface area. So really, when you look at a house, it's a pretty decent sized house of square footage. So about 27 to, somewhere around 3,000 square feet. So, when you really start thinking about that's a lot of surface area. So again, folds on top of folds on top of folds is why we get the surface area. So you have these valves of Endocrine or plicae circulares with villi on top of those, with these little microscopic folds on top of that with those microvilli. All these things are increasing surface area, and again, something that you want to absorb stuff through. The more surface area you have, the more capability you have to absorb, and that's why we have these folds on top of folds on top of folds. So you can see the plicae circulares on these images right here where the arrows are pointing at, those folds going up into it. We can see those on some of the slides that we'll look at in a little bit as well. And again, same basic layers here. You have the the mucosa. Again, simple columnar here. Still going to have the lamina propria. Still going to have that muscularis mucosa. We are then going to have a submucosa. Depending on which section we're in, we may see structures in there, we may not. And then we have the pretty standard muscularis. So the inner circular and the outer longitudinal layer. Again, nerve supply to this with that plexus again, those parts of that autonomic nervous system controlling it. And then we have that serosa that is deep to that. So you can see those little villi. Again, it's hard to see all this because, again, it all depends on the fold. So you can see here, we don't have just pieces of epithelium setting out in the lumen. That is actually with the section that we did, it happened to be in that plane where a villi was maybe coming into that field of view, but you don't see the bottom of it. So keep that in mind. And again, sometimes this may look like more than one layer. Again, it's depending on if you're getting a fold or other stuff like that when you're slicing into it. So these villi are these finger-like extensions that go out into the lumen. Again, more surface area, more ability to do that. We then have these glands that are in between these villi. They also have a fun name. We can call them intestinal glands, which isn't that much fun, or we can call them the crypts of Lieberkuhn. So we have valves of Kerkring and crypts of Lieberkuhn. Like I said, sounds like Dungeons and Dragons, something else like that. Kind of a funny names to this. But these are invaginations that go down in there. We'll have hormone-producing cells that are lining these, mucus-producing cells that are lining these. Again, epithelium covering going all the way down here. Sometimes there's going to be some immune cells down at the base of those as well. But different cell types, like I said, different parts. So you can see here's kind of showing you those villi going up, and you can see those crypts on either side of it. One of the things we'll see on a lot of these epithelial cells is there's going to be an intermixing of goblet cells. Generally, the further we get down the path to the anus, the more goblet cells we see. As we're getting to the end and like the descending colon into the rectal area, it's almost greater than 50% goblet cells. So as we absorb more and more liquid out, we start adding more and more mucus-producing cells to make sure everything keeps moving through the tube safely. So again, you can see these villi, and you can see how those crypts would work with those little invaginations going into that on each side of these villi. And if you actually look at this in a dissected animal, it almost has like a carpety structure to it, where you can kind of move it one way or the other, and you can see the kind of the villi there. It's hard to pick them out, each villi separately, but you can see that there. It's kind of like when you vacuum a carpet, all the fibers get going the same way. If you do that with the villi of the intestines, you can actually see that there's something standing up that you can kind of tip over or not. And again, you can see right here with those villi going up and those crypts going down in between. Again, mainly simple columnar cells have microvilli. A lot of times we talk about this being the brush border. These are making enzymes to help digest stuff, as well as really serving as these absorptive cells. The other thing we see mixed in there is goblet cells, like I was saying. Lots of mucus to keep this stuff moving along. And again, the further we get down the small intestine into the large intestine, the more goblet cells we see, because we're, again, absorbing more water out the further we go down. And you can kind of see this here, those just normal cells there, and the goblet cells with the lighter kind of look to them here. And those are the two main cell types that are in there. The other thing we see in there are these enteroendocrine cells. These make some hormones. Again, I'm not going to make you differentiate these, but these make hormones that are called CCK and secretin. These can act on the stomach, gallbladder, pancreas, and tell them to secrete their particular excretion, their substances that are going to be involved in digestion. At the bottom of these crypts, a lot of times, are what are going to be called Pantha cells. These make some antimicrobial substances, because again, the tube is really open to the outside when you think about it. So if we are going to get some type of pathogen from ingesting something, it is going to end up in the intestinal lumen. So it makes sense to have antibacterial substances being made and secreted into this lumen a little bit. And there's also some kind of immune macrophages there that are going to be ingesting this stuff and presenting them to immune cells. And again, much like throughout the digestive tract, there is a lot of, because it is open to the outside, there's a lot of damage that can happen here. So we do have a lot of stem cells that are continually replacing the intestinal lining. This is part of the reason if you have chemotherapy for some reason, the likelihood of having nausea and issues with digestion is a big thing, because there is so much turnover of that. Same thing with hair cells. Why you lose your hair when you do chemo, you're damaging those cells that are continually being replaced. And this, you can see some of that glandular epithelium as you get beneath this. And those would be those Pantha cells. Those little circular structures here, these kind of bright pinkish granular cells. Again, how we can tell this stuff apart, I already was kind of showing you with the stomach. You don't see the villi in the stomach, you do in here. You don't have Pantha cells in the stomach, you do in the small intestine. You have microvilli in the small intestine, you don't in the stomach. Those are some ways to tell it apart. Another area to look at is that submucosal area, which is one of the things I did point out to you already. Again, the duodenum, jejunum, and ileum, they are similar in that they have the same type of epithelium. They all have these crypts. They all have villi. They all have similar mucosal layers to them. Really, the big difference here is in that lamina propria and really more so in the submucosa. So the duodenum, it has in the submucosa these Brunner's glands. These are making an alkaline mucus that helps to neutralize, along with pancreatic secretions, that acidic kind that's coming in from the stomach. Only the duodenum has these Brunner's glands, and you can see them right in this image right here. There's your mucosal layer. Here is that Brunner's glands beneath that. I showed you that on that last slide, the stomach to duodenum slide. Again, only the duodenum has the Brunner's glands. And is this glandular epithelium, again, secreting alkaline mucus to neutralize the acidic kind. As we go to the tail end of this, we also have something that's distinct to the ileum. The ileum has what is called Peyer's patches. These are mucosally associated lymphatic tissue. It is lymphocytes in little groups in that submucosal layer. Again, makes sense. Anything we ingest could cause microbial infections. So having immune cells there that can trigger an immune response does make sense. And again, that is in the ileum. The ileum has that, and it makes it distinct. So you can see these immune cells in that ileum, and I will show you this on a slide on the tail end of this. The jejunum, what makes a jejunum special? Simple answer, nothing. It doesn't have Brunner's glands, it doesn't have Peyer's patches, and therefore it is distinct from the other two. Like it says there, sorry jejunum, you have nothing special about you, but you're special because you're not special. And I'm just joking around. But the main difference here, no Peyer's patches, no Brunner's glands, that which makes the jejunum distinct. So that's the villi, all the other stuff that we'd see in the intestine, but that submucosal layer does not have anything that makes it distinct. And you can kind of see this here. No Brunner's glands, no Peyer's patches, just a normal submucosal layer. And here's a nice little summary of these ones that kind of tells you this apart. You can see the different things here, what they have, what they don't have. And again, Brunner's glands distinct to the duodenum, Peyer's patches distinct to the ileum. The rest of them pretty similar. Goblet cells tend to increase as we go through this, so you can see some in the duodenum, more in the jejunum, most in the ileum. If we're not comparing it to the colon, it'll be more in the colon. So we'll take a look at some of the slides of this stuff, and then, like I said, we'll move on to the large intestine, and then that will be it for this one. Then we'll do the accessory glands in the next set of PowerPoints. All right, so we're looking at the same slide we were looking at the last time when I left off here. Again, just reminding everybody that if you were to zoom in on this, when you start looking at, went in and out. If you start looking at this, you can kind of see here is those internal structure, the lumen right here, your epithelium's right here, and these would be those Brunner's glands. And again, if we kind of move along this, you'll see that these Brunner's glands are present as we work down through the duodenum underneath that mucosal lining. If we move down here a little bit further, you can see a little bit of a plicae circularis. If I zoom it out just a little bit, you can see it kind of right here moving in this area right there. So that would be the duodenum. That is how I would differentiate it. If we go to the ileum next, this is the only good slide on all of our digital slide boxes I could find of an ileum. So you can see the lumen is right here. Here would be your mucosal lining. These structures along here would be those Peyer's patches. This is the only good one I could find. It was on the Histology, the Zoomify Histology from Loyola. It's this one right here, the ileum Peyer's patches. If I'm going to ask you this, I'll ask you that type of thing there. But again, being able to identify the different layers, definitely. So mucosal, submucosal, the muscularis, and the serosa would definitely be fair game. As you can see, this is the jejunum. You can see here are those little folds, those plicae circularis. You can see the mucosal lining right here. And again, you see in the submucosa that there is no submucosal structures other than just kind of your basic connective tissue. That's what we're looking at for each of these ones and how to differentiate them. Outside of that, knowing the cells, know what the intestinal crypts are called. So those would be those crypts of Lieberkühn. So something like that right here. We can actually zoom in on that a little bit. Probably get it right here. So those would be those crypts of Lieberkühn, or those intestinal crypts. And these are the little structures at the bottom here that the Pantha cells are going to be present inside of those. So if you look in these ones, you got these kind of pinkish cells here. Those would be those Pantha cells that are present at the bottom of those. So next time we'll look at the large intestine, and that will finish up our digestive tract.